This lecture will be a review of genetic screenings as well as the crime scene investigator lab. Initially, we were supposed to use the Bio-Rad kit. However, we will be instead using simulated experiments from PraxiLabs.com. These simulated experiments include the DNA extraction lab, polymerase chain reaction, and Agarose Gel electrophoresis. Followed by that, I will just review genetic screening techniques, which include DNA isolation, PCR, a restriction enzyme digest, Agarose Gel electrophoresis, DNA sequencing, and DNA fingerprinting. Followed by that, I will go over gel analysis and what documents you will need in order to complete that portion of the lab report. Just a review of the Bio-Rad kit. This kit allows you to run PCR and then complete an Agarose gel in order to determine which suspects you can exclude from a crime scene and which suspects cannot be excluded from a crime scene. This kit typically has 9 reagents that are contained within it. For example, there are 5 DNA samples, which include 4 suspects. That is suspects A, B, C, and D. In addition, you also get a crime scene suspect sample. Within these 9 reagents, you also receive loading dye, which is an orange solution in this case. You receive the molecular weight marker as well as PCR mix and primers. And using this PCR mix and primers, you're able to create a master mix in which you will aliquot within 5 PCR tubes as seen in the image on the left. So initially you would acquire 5 PCR tubes and aliquot into all 5 tubes the master mix and primers, which would have been pre-mixed. Into all those 5 PCR tubes, each student would have added each DNA sample. So one tube might receive the suspect A DNA sample, the second tube might receive the suspect B DNA sample, etc. Once all 5 tubes are complete, you would insert all 5 tubes into a thermocycler and run it for particular temperatures. This would allow your DNA to be amplified, which then you could run onto an Agarose gel, and then you would image that gel and analyze that gel. In this case, we are not able to utilize the Bio-Rad kit, so we will be utilizing PraxiLabs simulated experiments. For further information regarding the Bio-Rad kit, I have provided the link to the protocol. PraxiLabs originally allowed me to access 5 labs. However, a few weeks after creating my account, I did have access to a few different labs as well. However, for each student, if you do create an account, you will have access to the first experiment listed, which is DNA extraction, the third experiment listed, which is PCR, and the 4th experiment listed, which is electrophoresis. PraxiLabs will allow you to have a simulated experience for the DNA extraction as well as PCR and electrophoresis. And for the PCR conditions, I have included a screenshot on the bottom left corner of the screen. To review DNA isolation, this is a bit different than extraction. So for PraxiLabs, you do have a DNA extraction procedure. Isolation is a more thorough procedure which will allow you to isolate and purify the actual DNA molecule. And that is super important for running an adequate PCR and having a sufficient gel. For DNA isolation, we typically require several different buffers, a spin column, and cells. We do use the QIAGEN kit, the DNeasy QIAGEN kit, which includes all of these buffers, and it includes spin columns and collection tubes. Upon completion of the DNA isolation protocol, each sample should be quantified using a spectrophotometer. The spec that we utilize is the NanoDrop 2000. So pipetting a small amount A small volume of the DNA sample will allow you to measure DNA concentration, which is typically in nanograms per microliter. In addition, the NanoDrop will allow you to quantify how pure your DNA is. So if you do have a 260-280 ratio of 1.8, that will signify very pure DNA. In this image, you'll see the results from a NanoDrop. In this particular sample, the concentration is 11.7 nanograms per microliter, and the 260-280 ratio is 2.05. That tells me that this sample is not quite pure because it is not 1.8 regarding that ratio. For polymerase chain reaction, this is a technique used to amplify small amounts of DNA and create exponentially larger amounts of DNA, so millions and billions of copies. This procedure utilizes starting material, also known as template DNA, in order to produce larger amounts of that specific DNA region. PCR is typically used in forensic science and heavily in molecular biology. There are some terms I wanted to review prior to going into genetic screenings, and that is that a gene is a particular portion of a DNA sequence, which when this gene is expressed, it will code for a protein. Only protein coding genes code for proteins, however. A locus, this is the location at which a gene resides on a chromosome. The plural is loci. In the image on the right portion of the screen, you see two different alleles, and these are at one particular locus on both of the chromosomes. In this case, you see a red allele and a blue allele. This would signify a heterozygous genotype because each parent has contributed a different allele. And so the offspring would have a genotype which is heterozygous for that genotype. An allele, like I mentioned, is an alternative form of a gene. A heterozygous genotype encompasses two different alleles relating to a gene, as seen in the image to the right. A homozygous genotype, on the other hand, encompasses two identical alleles relating to a gene. And then the genome is basically all of the genes within one organism. And as you'll see in the other slides, Agarose Gel electrophoresis, this is a technique typically used after PCR, and this will allow you to separate DNA based on size. So for PCR, this protocol will require 2x master mix. We purchased this, however, it can be created in the lab. We also utilize diluted primers, which include forward and reverse primers, genomic DNA, which is our template DNA that we start with, sterile water, and a thermocycler. And the thermocycler runs at particular temperatures known as conditions or parameters. One of the temperatures is the annealing temperature, and this is very specific to the primers that are used. The forward primer, once it arrives, the tube will indicate on it the particular annealing temperature. So for example, a forward primer may have an annealing temp of 58 Celsius, and a reverse primer might have an annealing temp of 60 Celsius. And in that case, we would use 59 Celsius as our annealing temperature because it's a good medium between the annealing temp of the forward primer and the annealing temp of the reverse primer. And finally, PCR is typically used for DNA samples. For RNA amplification, you could utilize RT-PCR. The following image highlights the components for a PCR mix. You would start with 2x master mix. As I mentioned, we do purchase this. However, you could create it in the lab using nucleotides, buffers, and Taq polymerase. In addition to the master mix, we do need primers, the DNA sample, and one other component which is not pictured here, which is ultra pure water, and that will help dilute our 2x master mix down to 1x. Once we add the PCR tubes into the thermocycler, it will heat and cool in order to amplify our DNA molecules. So it will denature, anneal, and then extend. A particular set of PCR parameters that we commonly use in our lab starts off with 95 Celsius for 2 minutes. And then you go into the amplification cycle, which includes denaturing, annealing, and extending. So that is 95 Celsius for 30 seconds. The specific annealing temperature, which may be 58 Celsius for 30 seconds, and then 72 Celsius for 1 minute. And this runs for one cycle, and then it also repeats for 35 more cycles, which brings it to a total of 36 cycles in this case. Once the cycles have completed, you will move to 72 Celsius for 7 minutes, and then 4 Celsius forever, or for infinity, or until you stop the machine and freeze the samples. 4 Celsius is consistent with refrigerator temperatures. And then on the right, you see the PCR machine from Thermo Fisher. This includes 3 different blocks, so you could run samples that have different parameters at the same time. A restriction enzyme digest typically follows the PCR reaction, but not in all cases. For most genetic screens, you will not need a restriction enzyme digest, but if you do, it's good to know that this procedure will cut or cleave DNA at a specific SNP location. A SNP is a single nucleotide polymorphism, which indicates there's a nucleotide substitution. RE digests are typically used for some genetic screens, but not all genetic screens. In order to run this protocol, you will need your PCR product, a CutSmart buffer, a specific restriction enzyme, and a thermocycler, and typically you incubate it for a while prior to running your gel. Restriction enzymes are obtained from bacteria, and like I mentioned before, all RE digests are not required for all genetic screens. And then on the left, you see a chart. This is from New England BioLabs, and this encompasses all of the restriction enzymes that they have available for purchase. Agarose Gel electrophoresis is the next technique used in a genetic screen, and this method is used to separate bands of DNA based on size. DNA bands are typically measured in base pairs, also known as BP, and this technique requires agarose, TAE buffer, a gel electrophoresis box, loading dye, a PCR product, molecular weight marker, and a fluorescent stain. Gel red is a safe and non-toxic stain that we typically use. However, Ethidium bromide was used previously and is still used in some labs. Ethidium bromide is a carcinogen though. And just to mention, gel red and Ethidium bromide are both stains that can be added in small volumes to the melted agarose solution. So you can melt your agarose in TAE buffer, and then once it has turned into a clear solution, you can add a few microliters of gel red or a few microliters of Ethidium bromide, and then allow it to cool and then pour it into the casting tray. Once the electrophoresis is complete, you can image it and it will be ready to go. Another method is to not add the stain and to just melt the agarose, pour it once it cools, and then pipette your samples, run the electrophoresis, and then before imaging, you can stain it in an Ethidium bromide solution for about 10 or 15 minutes, followed by a destain in water for about 1 hour. However, that is not the most efficient way to stain a gel. Upon staining and upon completion of the electrophoresis protocol, you will need to image in a ChemiDoc, which is in the next slide. So, but within this slide, I have an image of Ethidium bromide, of gel red, and then various bands that have been stained by various different stains, including CyberSafe and gel green. And then there's also a voltage source from Bio-Rad. And typically we run gels for about 45 minutes at 100 volts. In the next slide, there is various images of electrophoresis chambers. On the very left, there's one chamber with the lid in addition to casting trays and combs. On the top right image, there's a gel that has already been cast and the TAE buffer has been added and the samples have been pipetted into the wells. In the bottom image, there's a stained gel, and then there's the photograph, which was taken after the imaging, which is typically black and white. In this image, you see a ChemiDoc, which is the imaging system that we utilize. And on the right, there's an image of a gel. So in the gel image, there's a molecular weight marker in the first well. There are 6 bands of DNA. And then the molecular weight marker is also visible on the last well because it was pipetted twice in this case. In this slide, I just want to outline the differences between an agarose gel that did not have a restriction enzyme digest versus a gel that did contain a digest. It contains digested PCR products. So in the gel that's visible on the left, you see a molecular weight marker and a band of DNA. It is just a regular band of DNA. It has not been cut with a restriction enzyme. On the gel on the right, you do have the molecular weight marker, but you also see 3 different samples. The topmost sample represents a homozygous genotype. This band did have a restriction enzyme digest added to it. However, the RE digest did not cut because it was not necessary. This particular sample represents a homozygous genotype because the one band would show up just one time, and this indicates that both parents contributed the same allele. Therefore, the allele shows up one time. So in this case, both parents contributed the same allele. The offspring has a homozygous genotype, so this individual has the same allele on both chromosomes for this particular gene. So there would be redundancy if this band showed up twice, but because there is no redundancy here, it needs to show up one time. And if it does show up one time, it indicates that both parents contributed the same allele. In the next sample, there are 2 different band structures. The first one is not cut, and the second one is cut. This indicates that one parent contributed a band which was not cut, and the other parent contributed a band which was cut. This indicates that this offspring, that this individual has a heterozygous genotype for this particular trait. In the third well, in the third sample, the band is cut, and this indicates that both parents contributed a cut band to the offspring. So this individual has a cut band on both chromosomes, for both alleles on both chromosomes. So it is the same cut. It shows up one time, although it is basically the same cut from both parents. So it would have shown up 2 times, except there is no redundancy here. It only needs to show up one time to tell me that both parents contributed the same allele. Therefore, this is homozygous. Followed by agarose gel electrophoresis. If DNA sequencing is required, there are intermediate steps between electrophoresis and getting the samples ready for sequencing. Once samples are ready for sequencing and they are inserted into the sequencer, the data which results will appear in a file that is basically a chromatogram. The chromatogram can be opened up using software such as 4Peaks on Mac or Chromas on PC. And you'll notice in the chromatogram, you have all of the nucleotides and then the peaks which correlate with those nucleotides. However, you'll see you have some single peaks, and then you have some peaks that are merged. So you might see a blue peak that is a sloppy peak. It should have been, for example, 3 distinctive blue peaks, but it has merged into one larger peak. This is typically not what you want to see. I also can observe that within the black peaks and the red peaks, there are 2 or 3 peaks which should have been distinct. However, they have become merged. This indicates there were some issues in the prep for the sequencing products. Typically, what you do want to see is single distinctive peaks. For example, in the image on the bottom right, this is from one of my sequencing products. All of these peaks are distinctive peaks, and they are relatively tall peaks. So just recall that you do need to have distinctive peaks with your DNA sequence chromatogram and not merged peaks. DNA fingerprinting is a much different protocol. For this, you typically don't utilize a PCR master mix. You just have your isolated DNA, and you add to that fingerprinting mix. Usually it can be identifiler or Y-filer mix, and then you will incubate that into a thermocycler, and once it's added to the sequencer, it can result with a DNA fingerprint. You will not have all of the peaks in this figure for your fingerprint. You might have some of them if you are doing this Y-filer protocol, but typically you will only have a few handful from each section. And for DNA fingerprinting, the software that we utilize is Peak Scanner for PC. Gel analysis will be the next topic. So assuming that each student has gone through the PraxiLabs simulations, which include DNA extraction, PCR, and gel electrophoresis, you'll be able to download the gel photograph, the log paper, and the crime scene analysis instructions and worksheets from Blackboard. I have scanned 2 different gels, Gel 1 and Gel 2. Depending on your last name, you will need to download one of the 2. You can also download the log paper and the instructions and worksheet. It is recommended that you do print these documents. However, if you do not have access to a printer, I'm totally fine with completing this analysis on blank sheets of paper and doing it manually. So to review, the gel photograph will need to be labeled, the log paper will need to be completed, and the worksheet will need to be completed, and all 3 of these documents will need to be inserted into your lab report. So for the instructions, it mentions that you will need to print your own gel image, but if you do not have access to a printer, I'm fine if these are done by hand. The first thing you'll need to do is draw a single vertical line or a point of origin on your printed gel image. I have a point of origin drawn here as an example, but let me go forward to the gel images. I have drawn a makeshift point of origin. This just is something I did for this presentation. However, you will need to draw your own point of origin on your gel. Once you do that, you will need to take some measurements. So step 2, on your gel, label all 5 samples and also the molecular weight marker. I've already done this for both of the gels. So I've already labeled the crime scene as well as suspects A through D, as well as the molecular weight marker on both gels. Each student will need to draw the point of origin and will need to label the molecular weight marker. So what I mean by that is the second part of step 2, use the blank space to label all of the base pair amounts for that molecular weight marker. This is pretty straightforward because I've already done it on this particular slide. So it starts at 1,500 base pairs, and then the next band indicates 1,000 base pairs. The third band indicates 700 base pairs, and then 500 base pairs all the way down to 100 base pairs. So the band at the very top on both of these gels for the marker indicates 1,500 base pairs. This is because it's a relatively large band and it does not move that far down the gel. Whereas the smallest band in the marker, which is 100 base pairs, has moved all the way down the gel and it is the last band within the marker on both gels. So once that's completed, you'll need to proceed to step 3, which indicates that you need to complete part 1 of the worksheet. So part 1 of the worksheet entails measurements of the molecular weight marker bands. For this, you'll need a centimeter ruler, or you could possibly use a virtual ruler if you'd like to complete this on your computer. You'll need to measure from the point of origin to the first band of the marker. So if I'm going to use, for example, the gel on the left, I'm going to move to the marker on the gel on the left, gel 1. From the blue line, I will measure to the first band of the marker, which is the 1500 base pair band. And I will write down a certain length in centimeters. And I've actually already done this. For the first band, I measured 2.8 centimeters. And you can see I did complete the measurements for all 8 bands. And for example, for band 8 of the molecular weight marker, I measured 6.3 centimeters. So going back on the gel on the left, gel 1, from the blue point of origin to the last band of the marker, which is the bottommost band, I did measure 6.3 centimeters. And that was using this particular gel, which I had printed out, and I had used an actual centimeter ruler to do this. Once step 1 is completed, the molecular weight marker band measurements, you'll need to go back to the instructions. So step 4 mentions using your measurements from part 1, graph a standard curve. So these are my measurements. Band 1 is 2.8 centimeters against 1500 BP. So I used log paper, as seen on the right, and here's a zoomed in image of it. I used log paper to graph my standard curve. But the first thing I did was label the x-axis 1 through 7. And this would indicate distance migrated in centimeters. These are basically the band measurements. So for every 10 boxes, I labeled in intervals of 1. So every 10 boxes, I labeled 1 centimeter, and then 10 more boxes over, I labeled as 2 centimeters, all the way until 7 centimeters. On the y-axis, I labeled the length in base pairs. So the length of my bands of DNA. This might seem confusing how I labeled the BP amounts, but I basically started on the very bottom left corner of this log paper where it mentions semi-logarithmic cycles. There's a tiny little 1 on the left, and that to me indicates 100 BP. And then there was a jump to a 2, which indicates 200 BP. And then a 3, a 4, a 5, all the way up to 9, which indicates 900 base pairs. And then also another 1. So for this number 1, I drew a line and I wrote 1000 BP. And then it jumps to a 2, which could be 2000 BP. However, I do not need a line which indicates 2000 base pairs. So I drew a line between the 1 and the 2, and I labeled this as 1500 base pairs. And also, just for fun, I labeled the very top one as 10,000 BP. Once I did this, I went back to step 4 of the instructions. So using the measurements from part 1, graph a standard curve, and I graphed a curve. And so you'll notice for all 8 bands, I have 8 little dots, and I drew a line through it, and this was my standard curve. So for the first point on my curve, this was for the first band, which was 2.8 centimeters against 1500 base pairs. So on the x-axis, I went from 1 to 2 to 2.8, which was 8 boxes to the right. And I followed this up to my 1500 BP line, and I made a little point. Then I moved on to the second band, which was 3.5 centimeters against 1000 BP. Again, on the x-axis, I went down to the 3 to 3.5, which is directly in the middle of 3 and 4, and I moved upwards to 1000 base pairs, and I made a point. I repeated this for bands 3 through 8. Once I did that, I had completed my curve, and then I drew a line through the curve. So you can ignore the little highlighted marks for right now, but there are 8 different points on this standard curve. And then I went back to the instructions. Step 5 indicates that on the worksheet, you should complete part 2. So part 2 entails the band measurements, all 10 band measurements that were from the suspect samples and the crime scene sample. So I went back to my labeled gel and I measured from the point of origin to the first band of the crime scene sample. I also measured from the point of origin to the second band of the crime scene sample, and I inputted that in part 2. So for the crime scene sample, I did measure 4.6 centimeters and 6.1 centimeters. And then I completed this for the entire section of part 2, just repeating these measurements until I was complete. So going back to the instructions, step 6 mentions graph the measurements from part 2 onto the semi-log paper. And this is why I had some highlighted portions on the standard curve, because there was a lot of overlap. For example, the first band for the crime scene was 4.6 centimeters. So I went down onto the x-axis and I found where 4.6 centimeters was. So between 4 and 5 is 4.5, and I moved one box to the right, which is 4.6 centimeters. I followed that up where it merged with the standard curve plot line, and then I kind of looked to the left. I followed it all the way to the left and saw it was near about 500 BP. It's easier to do this on a physical copy though. So for the 4.6 centimeters, I determined that it was 500 base pairs. For the second band, this was 6.1 centimeters. I did the same thing. So I found 6.1 centimeters on the x-axis and I followed it up and I noticed it was near 300 to 400 base pairs. So using a physical document in this case is super helpful. But for the second band of the crime scene, I did measure it as 130 base pairs. So yeah, I think I misspoke. So in this case, 6.1 does correlate with 130 base pairs. So the one that is on the bottom left corner of the graph, that is 100 base pairs, and it jumps to 200 base pairs. Therefore, each box in this case between the 1 and the 2 on the left will be 10 base pairs. So that is 130 base pairs for the second band of the crime scene. So I repeated this for all 10 bands for part 3. And going back to the instructions, on questions on part 7 on your worksheet, it mentions to complete part 3. This has already been completed. Here you'll determine the base pair amounts, which we have, and there will be 2 base pair amounts per suspect sample. So once I have all of the suspect samples and all of the base pair amounts highlighted in part 3, I can go through and compare them. So the crime scene, the first band, there are 500 base pairs, and the second band has a length of 130 base pairs. So suspect D, I can exclude because 1,000 base pairs is not at all similar to the crime scene band, which has 500 base pairs. So in this case, I can exclude suspect D. Going to suspect A, I can also exclude suspect A, which has 840 base pairs for the first band. And that is not similar at all to the crime scene suspect band, which is 500 base pairs. Suspect B and C, however, I cannot exclude because although they are similar to the crime scene, there's not that much of a deviation for me to exclude them. So going back to the gel image, suspect B and suspect C are quite similar to the crime scene. So in this case, I could exclude suspect D and suspect A, but suspects B and C still remain as suspects. And so here I have a labeled gel, just as an example. For this particular experiment, you will have each labeled your gels by hand, or you might decide to label them using a computer. However, I did want to outline what is appropriate and what is not appropriate when labeling a gel. So this gel on this particular slide is very informative. It includes the DNA ladder on the right, also known as a molecular weight marker. It indicates the samples, and it also indicates cut sites. So this gel is a gel which did require a restriction enzyme digest prior. There are some things about this gel that would not be recommended. So for example, although it is very informative, I would not use arrows or lines on a gel. I would also not make markings or annotations directly on the gel. I would keep them on the exterior of the gel. So maybe above the wells and to the right of where the marker is, something like that. So this would be sufficient because it does not directly annotate on top of the gel, but actually away from the data. So just as an overview, the labeled gel, the completed log paper, and the completed worksheet will need to be inserted into the results section of the crime scene lab report. And that concludes this presentation.